project is the effects of carbon dioxide on freshwater plants.
We hypothesized that higher amounts of sodium bicarbonate will increase the oxygen production in the freshwater plants.
We conducted this experiment by measuring levels of dissolved oxygen in response to varying levels of baking soda, or sodium bicarbonate. We use baking soda because it raises carbon dioxide levels in the water with the goal of improving photosynthesis. We used 8 freshwater aniceris plants and divided them into 4 groups. The control is the 2 plants that received 0 grams of baking soda, and the other 3 groups contained 0.5, 1, and 2 grams of baking soda. The independent variable is the amount of baking soda in each container, and the dependent variable is the level of dissolved oxygen. Okay, so here are our graphs and data tables just to go over some numbers. Week 1 in our data table of plants number 8 with 2 grams and plant number 7 with 2 grams, they both have 18 and 19.9 milligrams per liter of dissolved oxygen, which is much different than the 12 and 11.5 milligrams in plants 1 and 2. As shown in the graph, the plants with higher sodium bicarbonate levels immediately produced higher amounts of dissolved oxygen.
Our experiment was conducted by preparing 8 identical containers filled with 1 liter of tap water each. Our first group was the control, and they had 0 grams of baking soda. Our second group was labeled 3 and 4, and they each had half a gram of baking soda. And 1 gram of baking soda was added to containers 5 and 6. Finally, 2 grams of baking soda was added to containers 7 and 8. And we used an aniceris plant, and the plants were exposed to the same conditions in the same spot in my sunroom. And we also used an oxygen probe to measure the dissolved oxygen levels of the plants in each container. Across our 7-day testing period, the oxygen levels across all groups declined. This is likely due to an supersaturation period due to the increase in sodium bicarbonate levels available to the plant and an increase in pH levels. The group with 2 grams showed the sharpest decline, with early bubbling and supersaturation, which was then followed by poor plant health and low oxygen levels, possibly due to pH imbalances. The 0.5 grams and 1 gram groups performed the best overall. This was shown by their balanced oxygen output and sustained plant health, which supported the idea that moderate carbon dioxide levels enhance photosynthesis without negative side effects. These findings help show the significance of optimal CO2 levels in aquatic environments. Similarly, in a study done by Chao Zhang in PubMed, they similarly found that increased CO2 concentration influenced the growth and nutrient content of plants in Tahoe Lake. If me and my partner Michael were to do this experiment again, we would have increased the number of plants to 9, but reduced the number of groups to 3, having 3 in each group.
To higher levels of dissolved oxygen, showing that there was more carbon dioxide availability. The results partially support the hypothesis, which states that the higher amounts of sodium bicarbonate would increase oxygen production by enhancing photosynthesis. The plants with 2 grams of baking soda produced more dissolved oxygen, but quickly decayed and showed signs of stress. But however, plants given moderate amounts of baking soda, such as 0.5 and 1 gram, maintained healthier and more consistent levels of dissolved oxygen and still kept a strong physical appearance. This suggests that while adding carbon dioxide may boost photosynthesis, excessive levels may be harmful. We also